شو أخباركم يا شباب؟
والله الحمد لله بخير.
شكل الحضور قليل. عندكم اختبار قريب؟
الأسبوع الجاي.
وش اختبار ايش؟
فاينل GND.
صعبة. صعبة المادة عليكم؟
والله إن شاء الله أنها مقدور عليها، بس لأنه وراها HP فشوي يعني الوضع.
مثل HP.
ال HP هو الأسبوع الجاي GND، بعدها عندنا اختبار ثاني مادة ثانية
أها.
Learning Skills، وبعدها ال HP.
لأنه ال HP يتوقع باقي أسبوعين أو أكثر حتى على اختبار ال HP، لأن في عندكم ريفيجن معي. صح؟
كويس.
اللي يعرفه أنا ما أدري، تراهم يغيرون الجداول والله، يعني كل أسبوع ترى في جدول مختلف. فما أدري بالضبط. يعني اللي أعرف أن عندي ريفيجن معكم مو بالأسبوع الجاي، الأسبوع اللي بعده.
مم.
فأتوقع أن الاختبار منطقيا بيكون بعد الريفيجن، ما راح يكون قبل الريفيجن.
اي صحيح.
آه طيب. عددكم انتم 113 ولا؟
إيه تقريبا.
اوكي، شوي بس ننتظر لين صالون 70 وبعدين نبدأ إن شاء الله. طيب، على بال ما يجون الشباب، اليوم محاضرتنا بتكون ميكس من أشياء كثيرة أنتم أخذتوها سابقا. تقريبا 90% من دون مبالغة، 90% من المعلومات اللي بتنقال في المحاضرة هذه، خلينا عشان ما أبالغ، لكن 70% من المعلومات اللي بتنقال في المحاضرة هذه هي معلومات كده أخذتوها ومرت عليكم خلال المحاضرات الماضية معي أو مع الزملاء الثانيين اللي يشرحوا لكم، الدكتور صلاح. طيب، احنا عرفنا الجلايكوليسيس. وش الجلايكوليسيس؟
بريك داون اوف جلوكوز.
بريك داون اوف جلوكوز. ممتاز. الجلايكوليسيس في معها عدة بروسيس متصلة بالجلايكوليسيس هذه. أمس أخذنا بروسيس اللي هي البايروفيت لأستايل كو اي. وش اسم البروسيس هذه؟
ديكاربوكسيليشن مدري والله نسيت.
ديكاربوكسيليشن، بايروفيت
أيوة.
ديكاربوكسيليشن.
أيوة.
تمام. الآن لما تكلمنا عن تعرفون الماليت بروسيس كيف يطلع؟ تعرفون انه في نهايه التي سي اي سايكل راح يتكون اوكسالو اسيتيت. تمام. طيب، هل اخذتم اي شيء عن تحول البايروفيت لاوكسالو اسيتيت قبل في المحاضرات الماضيه؟
هو TCA ما
بس أخذنا المالت شنت والشنت الثاني بس.
ممتاز.
أخذنا TCA.
الماليت شنت موجود فيها الاوكسالو اسيتيت صح ولا لا؟
إيه.
إيه نعم.
ممتاز. احد منكم يقدر يذكرني بالبروسيس اللي من البايروفيت لين الماليت؟ وش الفكره؟ ليش الماليت شنت؟ وش فكره الماليت شنت؟
علشان اللي هو بعض فكرة الماليت شنت إنه في بعض الناد لم يكون برا الميتوكوندريا، فما
ممتاز.
ما يقدر يدخل، يحتاج الماليت شنت أو هذا.
ممتاز. هل البايروفيت له دخل في الماليت شنت؟
إيه له دخل.
شلون؟
إنه شو اسمه، لا إله إلا الله. في شو اسمه اللي هو البايرود ديكار اللي هو فيه دخل بالتي سي اي، انه
مه.
بعدين بيصير لما بعدين السكشنال كو اي يصير
مه.
بعد سكشنيت، بعد فيوميريت، بعد يصير
مه.
ماليت.
مه.
طبعا هذه ما اخذناها، بس يعني يوم جيت لفيت لفه على ليبن كوت.
اوكي اوكي اوكي. ممتاز.
طيب تصير ريدكشن فتصير أوكزالو أسيتيت عشان يصير مالت، والمالت يشيل النات جوا الميتوكوندريا.
اوكي اوكي تمام. يعني احنا عارفين ان الماليت شنت تكون في الميتوكوندريا وخارج الميتوكوندريا، صح ولا لا؟
ايه هي
يعني الماليت يعتبر كسبستريت للعبور او انترميديت لعبور ايش الالكترونات، صح؟ او لعبور رسائل من داخل الميتوكوندريا لخارج الميتوكوندريا. زي مثلا الاوكسالو اسيتيت ما يقدر يطلع من الميتوكوندريا، وش يصير فيه؟ يتحول لماليت، يطلع خارج الميتوكوندريا، يرجع مره ثانيه الاوكسالو اسيتيت.
دكتور عفوا، هذه اللي نفسها الماريت شاتل. هي يعني وش الفرق بين الشنت والشاتل؟
الماليت شاتل والماليت شنت، والله حاليا ما تحضرني الآن. أنا أذكر شرحتها للطلاب السنة الماضية، لكن صار في تغيير فيها، لكنها ما تحضرني الآن. فخل سؤالك هذا شوي أجاوبه لكم في الريفيجن إذا هو السؤال جدا مهم يعني. تمام؟
تمام.
طيب نبدأ محاضرتنا اليوم. We will be talking about gluconeogenesis, the definition of gluconeogenesis, identify the importance of gluconeogenesis as important pathway for glucose production, describe the main reactions of gluconeogenesis, identify the rate-limiting enzymes of gluconeogenesis, and the gluconeogenesis as energy-consuming process, anabolic pathway, and able to calculate the energy consuming during the gluconeogenesis. So basically, this is an overview. Okay. In general, gluconeogenesis is the synthesis of glucose from non-carbohydrate sources. So if we think about non-carbohydrate sources that can be used to synthesize glucose, can you give me 3 non-carbohydrate sources?
Amino acids.
Amino acids, yes.
Lipids.
Lipids, yes.
Fatty acids.
Well, not all fatty acids. Okay. Lipid, it can be lipid. Why? There are 2 types of lipid, can be a source of glucose. We will talk about them today. And what else? There is something that's very, very obvious, and
Lactate, صح؟
lactate, exactly. Lactate. So all of these, I see someone wrote to me pyruvate. All of these molecules will have to go back to pyruvate, and then they go back to synthesize their gluconeogenesis. But what we need to think about is that the phosphoenol pyruvate to pyruvate is irreversible step. So the pyruvate has to make a detour in order to get away of that step, isn't it? So we need to think about this. We have mainly gluconeogenesis is not exactly reverse to the glycolysis. However, it's almost the same except in 3 steps, which are the irreversible step. Can anyone remind me by the 3 irreversible steps?
Glucokinase. And hexokinase also.
Hexokinase and glucokinase step, which is the first step, yes. What's the other step?
Phosphofructokinase.
Phosphofructokinase, yes.
Pyruvate kinase.
Pyruvate kinase. So these are the 3 irreversible steps. We go back to the gluconeogenesis. Gluconeogenesis principally or mainly take place in liver, and sometimes or by 10% happens in kidney. During overnight fasting, there will be up to 50% of consuming of the gluconeogenesis. So the gluconeogenesis will be very important while we are sleeping or while we are fasting for a very long time. So it occurs in both cytosol and mitochondria. And why is that? Because these molecules will go back to pyruvate, and then the pyruvate will have to travel to the mitochondria in order to change to oxaloacetate, and the oxaloacetate will change to malate. The malate will be transferring to the cytosol again, will change to oxaloacetate outside in the cytosol, and then from the oxaloacetate, we will build the phosphoenol pyruvate. This we will see in the next few slides. So what's the importance of gluconeogenesis? The importance of gluconeogenesis is they are the source of the blood glucose during the fasting for more than 18 hours, and for energy requirements in the brain, red blood cell, exercising muscles, and the renal medulla, cornea, and islands. So basically, for normal people, the blood glucose will go up to around 150, okay, when we eat a meal, it will go up around 150 or above that a little bit, and then it will go back again to the normal level, which is between the 80 to 100, okay? And during being in the 80 or between 80 and 100, this level of glucose will be supported by gluconeogenesis, okay? This will be supported by gluconeogenesis and glycolysis and glycogenolysis. We will talk later after your first exam. I think we will be talking about glycogen synthesis. So don't worry about it. The gluconeogenesis, we'll be talking about it today. So what are the substrate of gluconeogenesis? You guys have already mentioned some of the substrate, and the first one of them, the most important one, is the lactate. So what are the source of the lactate inside the body? We know that the glycolysis, the end product of glycolysis, is pyruvate, and under anaerobic condition in the muscles, when this person is exercising, walking, or something like that, the level of oxygen will go down, and then the pyruvate will start changing to lactate. Also, we know that the environment in the red blood cells is anaerobic, and always the product of pyruvate is lactate. So increasing the lactate inside the body will send a signal to the body that the body needs more glucose, and the muscles are missing glucose and need energy. So this lactate will go to the liver. As we mentioned, the synthesis of gluconeogenesis take place in two organs, liver and kidney. That's very important to know, okay? Gluconeogenesis does not take place in muscles, does not take place in any other part of the body except the liver and the kidney, okay? So the lactate is released into the blood by exercising skeletal muscles and by red blood cells. And this process, when the lactate released in the blood and go to the kidney and resynthesize the glucose in the, the kidney or the liver, and resynthesize the glucose, this process called gluconeogenesis. And this cycle, we call this cycle Cori cycle. So the Cori cycle represents the travel of the lactate to the liver or the kidney in order to synthesize glucose. So the entry of lactate. Lactate is formed by active skeletal muscles when the rate of glycolysis exceeds the rate of oxidative metabolism. During the anaerobic glycolysis in skeletal muscles, the pyruvate is reduced to lactate by lactate dehydrogenase. This reaction serves two critical functions during anaerobic glycolysis. So we already mentioned one of them. So why the pyruvate changed to lactate? How this will help the muscle or the red blood cell? What it will provide?
It's provide energy for the blood cells
Energy without oxygen.
because there is no mitochondria, no oxygen.
Okay, how? Well, it's not providing energy. Let's think. We mentioned that there are the NAD, which is the oxidizing agent, will be reduced during the glycolysis, yes?
Yes.
And we mentioned that in order to change pyruvate to lactic acid in the presence of lactate dehydrogenase enzyme, there will be an oxidation for this reduced NAD, isn't it?
Yes.
Yes.
Okay. There is a very important information about NAD. what is it? I have mentioned it several times that the level of NAD is limited in the cytoplasm and also limited in the mitochondria. Also, the NAD cannot travel from the mitochondria to the cytoplasm, and also the other way around. The one in the cytoplasm cannot travel to mitochondria. So we have a limited amount of oxidizing agent NAD inside the cytosol. So in order for the glycolysis to take place again, this reduced NAD has to be
Oxidized.
reoxidized. How it can be reoxidized in the red blood cells and the anaerobic skeletal muscles?
By lactate.
Lactate, pyruvate to lactate.
Yeah, by changing pyruvate to lactate. Okay, so this is one benefit of the synthesizing of the lactate. Another thing is that lactate produced by lactate dehydrogenase reaction is released to the bloodstream and transported to liver, where it's converted to glucose. The glucose is then returned to the blood for use by the muscles as energy source and to replenish glycogen storage. This cycle is termed the Cori cycle. Okay, you have to know what does this mean, and what the name of this cycle is the Cori cycle. Okay.
Okay.
So this is a figure just to explain the Cori cycle. We have high activity, strong ATP demand in the muscles. This would lead to hydrolysis of glycogen and glucose. And then at the end, we will have a lot of lactate because there is a shortage in the glucose. Therefore, the lactate will travel to the liver where there is sufficient oxygen and ATP, where the lactate will actually change to glucose. This process called gluconeogenesis, and the process of degradation for glucose and glycogen calls glycogenolysis and glycolysis. Okay? Which call, and in general, this cycle called Cori cycle. So, one part of the lipid is the glycerol. Why the glycerol is important for the lipid? How the lipid use glycerol? Guys, I have explained the lipids for you last year. That make me a little bit disappointed.
To connect with the fatty acids?
Yes.
Okay, and synthesize what?
Triacylglycerol.
Triacylglycerol. Great. So we will know that the hydrolysis of the triacylglycerol in the lipolysis will cause the production of glycerol. Okay. So how this glycerol is important for the body? In the kidney, there is a specific enzyme that's only present in the kidney, which is the glycerol kinase. When there is a hydrolysis for the triacylglycerol, and we get the glycerol, it will be phosphorylated, and an ATP will be consumed for this phosphorylation. This will give us the glycerol 3-phosphate. And we know that the glycerol 3-phosphate is an intermediate for glycolysis. Later, the glycerol 3-phosphate will be changing into dihydroxyacetone phosphate. And from the dihydroxyacetone phosphate, the fructose or 1,6 fructose bisphosphate will be synthesized. Okay, so we know that how the 1,6 fructose bisphosphate is synthesized from dihydroxyacetone phosphate. Okay, the glycerol 3-phosphate is an intermediate molecule in this reaction, but the glycerol 3-phosphate cannot directly go to synthesize the by itself to synthesize the fructose 1,6 bisphosphate. It has to also synthesize the dihydroxyacetone phosphate. Okay.
Okay.
This is just a figure for the reaction. This is just a figure for the reaction to explain fat degradation, glycerol. The glycerol will be via glycerol kinase will give us glycerol 3-phosphate, and the glycerol 3-phosphate, it will have 2 ways in order to synthesize the dihydroxyacetone phosphate. What is this way? The way that it has to pass through the mitochondria. What do we call this way? We have 2 glycerol 3-phosphate dehydrogenase, enzyme 1 and enzyme 2. Enzyme 2 is present in between the outer and the inner mitochondria membrane, and we know that there is a glycerol 3-phosphate shuttle, isn't it? It's different than the aspartate malate shuttle. Do you remember this? And it has another glycerol 3-phosphate dehydrogenase complex. So I will not take you to this in deep, but you should know that it has 2 ways to produce dihydroxyacetone phosphate, and the dihydroxyacetone phosphate is the key to synthesize the 1,6-fructose bisphosphate. The second thing is the third substrate is the amino acid. So the glucogenic amino acid derived from hydrolysis of tissue protein are the major source of glucose during fasting after the liver glycogen is depleted. The glycogenic amino acid are deaminated, giving pyruvate or intermediates of Krebs cycle. So it's direct, either the amino acid or glucogenic amino acid directly being deaminated into pyruvate, or it will change to an intermediate of the Krebs cycle. I will show you that the Krebs cycle in general and what are the intermediates of amino acid that can be involved in the Krebs cycle, all of which can be transformed into oxaloacetate, which is a direct precursor of phosphoenolpyruvate. Okay, all amino acids can give glucose except leucine and lysine, and we will know because leucine and lysine will change to pyruvate, and then from pyruvate they will form ketone bodies. They will not go to the Krebs cycle and form oxaloacetate. This is why they are not involved in synthesizing of the glucose. Glucogenic amino acid. When we say glucogenic amino acid, we mean that amino acid that's involved in synthesizing of glucose. So for example, leucine and lysine, they are not glucogenic amino acid, they are ketogenic amino acid because they will synthesize the ketone body. So amino acid breakdown to form glucose, the amino acid derived from dietary proteins, tissue protein, or from the breakdown of skeletal muscles protein during starvation. After transamination or deamination, glucogenic amino acid yield either pyruvate or intermediates of the citric acid cycle or Krebs cycle. Amino acid that are degraded to pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, or oxaloacetate are termed glucogenic amino acid. Amino acids that are degraded to acetyl-CoA are termed ketogenic amino acid because they can give rise to ketone bodies or fatty acid. Okay. Oh, sorry. So this actually is the general description for what's happening. So we see that some of the amino acid, they can directly change to pyruvate. Some of the amino acid can change to acetyl-CoA, and some of the amino acid, they can only change to acetoacetyl-CoA. Okay, we see that leucine and lysine, they are both involved in forming acetyl-CoA, and both of them, they cannot go inside the Krebs cycle because they will synthesize the ketone bodies. However, the rest of the amino acid, all of them can go to form acetyl-CoA. Okay, no, sorry, all of them can go and form oxaloacetate, which will be later used for glucose synthesis. So in here in this table, we have design it into 3 types of amino acid: the non-essential amino acid and the essential amino acid. Which one are only glucogenic? Which one can be glucogenic and ketogenic? And which one that can be only ketogenic, which cannot be involved in glucose synthesis? So glucogenic and ketogenic, both of these tables can be used in synthesizing of glucose. However, the ketogenic table, which is leucine and lysine, cannot be used in synthesizing of glucose. So the entry of glucogenic amino acid, the alanine, serine, cysteine, glycine, threonine, and tryptophan can be converted to pyruvate directly to produce glucose. The aspartate and asparagine can be converted to oxaloacetate. The alpha-ketoglutarate is the point of entry of several 5-carbon amino acids that are first converted to glutamate. And this, we will see the importance of this in synthesizing of alanine, and then this alanine will travel to the liver. I have mentioned that this alanine will travel to the liver, and in the liver, this alanine will be involved in releasing the non-essential amino acid to go to the urea cycle, and also will produce a pyruvate, which will be used later in gluconeogenesis. Succinyl-CoA is a point of entry for some of the carbon atoms: methionine, isoleucine, and valine. Propenyl-CoA, and then the methyl mylonyl CoA are also intermediates in the breakdown of these 3 nonpolar amino acids. The fumarate is the point of entry of phenylalanine and tyrosine. I think these amino acids are very important as an examples for the type of amino acid that's involved in each of the Krebs cycle. It might get easier with you after knowing the Krebs cycle in depth, but still it's very important to know it. It's very important to know the difference between the ketogenic amino acid and the glucogenic amino acid. The 4th substrate, we mentioned that the fatty acid, and when I told you about the fatty acid, I told you not all fatty acid can be involved in synthesizing of glucose. For example, the even chain fatty acid can be only beta oxidized into forming acetyl-CoA. However, this acetyl-CoA cannot convert back to pyruvate. Therefore, even chain fatty acid cannot be involved in gluconeogenesis. Okay. However, the odd fatty acid chain will provide or will produce propenyl-CoA, and we know that propenyl-CoA will be oxidation later and converted to succinyl-CoA, and then it will work in the gluconeogenesis substrate. Therefore, the fats can contribute to gluconeogenesis and form glucose except even chain fatty acid. So you have to know that the even chain fatty acid cannot be involved or cannot be a substrate for pyruvate or for the gluconeogenesis, because they will change to acetyl-CoA, and then at that moment they cannot change to pyruvate. So the reaction that's unique for the gluconeogenesis, we mentioned that there are 3 irreversible reactions. These irreversible reactions include glucokinase or hexokinase, phosphofructokinase, and pyruvate kinase, which are irreversible and require 4 enzymes unique to gluconeogenesis for their reversal. So there are 4 enzymes needed to get away of these 3 irreversible steps. First, we will talk about the first 2 enzymes, which are the pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Okay, why we are using these 2 enzymes? So we know that in the pyruvate, inside the mitochondria, there will be a pyruvate. However, this pyruvate need to be carboxylated. What does it mean of carboxylation?
Adding of carbon.
Adding carbon. Do you remember yesterday when I told you removing carbon, removing CO2 or carboxyl group, what advantage that will be given of removing CO2? What is the advantage of that? Do you remember there was 2 extra electron came on, and this electron later, pardon me?
When we move the carbon, we add
There-
2 hydrogen.
2 hydrogen. That's mean we added electron. This electron will be donated later to the NAD, and the NAD will use them to produce energy. Yeah?
Yes.
Okay, now we are adding CO2. What does that mean? It is the reverse, as we talked yesterday. When removing CO2 will give us energy. However, adding CO2 will cost the body energy. Therefore, an ATP needs to be used because it has to activate the CO2 before the CO2 added to the pyruvate. Okay? Therefore, this step is a consuming energy step. The biotin is a very important in order to help for the activation of the CO2 via the ATP. Okay, and both of them are considered to be the cofactor and coenzymes of the pyruvate carboxylase. Then we will have the oxaloacetate. So the oxaloacetate will be stuck inside the mitochondria. Okay, however, in order for the oxaloacetate, it has to get a ticket, or it has to be changed. It has to be get some help in order to go outside of the mitochondria. So how that will happen? Oxaloacetate will be dehydrogenated via the malate dehydrogenase in order to form malate. Also, this process will cause the oxidation of the NAD. Okay, this process will cause the oxidation of the NAD. So now we will have oxidized NAD that cannot provide energy. This malate will have a specific gate on the mitochondria, will travel outside of the mitochondria, and then it will compensate. How it will compensate? It will produce, and it will be oxidized to form oxaloacetate. Okay, how it will be oxidized? By the reduction of NAD, by helping of NAD. The NAD will be outside, the NAD in the cytosol will be reduced, and this will allow for the oxidation of malate via the malate dehydrogenase enzyme, the help of malate dehydrogenase enzyme, and then we will form oxaloacetate. So now the pyruvate has changed to oxaloacetate. So we finished the first step, or we finished the benefit of the first enzyme, which is the pyruvate carboxylase. Now the oxaloacetate need to be changed to phosphoenol pyruvate. So this phosphoenol pyruvate, we need to use a specific enzyme which called phosphoenol pyruvate carboxylase kinase, or carboxykinase. So this enzyme will remove a CO2. Unfortunately, in this process, another ATP will be consumed, which is not ATP, GTP, which is the guanine triphosphate, will donate one phosphate, and at this process we will form the pyruvate phosphoenol, pyruvate. Okay, so these are the steps when pyruvate changed to phosphoenol pyruvate. There are 2 enzymes involved: the pyruvate carboxylase or phosphoenol pyruvate carboxykinase. Let me check. Yeah, phosphoenol pyruvate carboxykinase. So what's the benefit of this reaction and that take place in the mitochondria and the kidney, first to allow to produce the phosphoenol pyruvate, which is an important substrate for the gluconeogenesis, also to provide oxaloacetate that can replenish the TCA cycle intermediate that may become depleted. So the oxaloacetate can directly compensate for the elements or the intermediates of the TCA cycle that depleted during the loss of energy and the decrease of glucose. So muscle cells also contain the PC, but which is the pyruvate kinase, but use the oxaloacetate product only for replenishment purposes. And do not synthesize glucose, okay? So there is no glucose synthesis in the muscles, and we will know later because there is an enzyme that's not present in the muscles. It's only present in the liver. Therefore, there is no purpose for the muscles to synthesize glucose. The second reversal step, which is the phosphofructokinase reaction. We know that this phosphofructokinase reaction was a very, very complicated one, isn't it?
Yeah.
So can anyone remind me of it?
We need dihydroxyacetone phosphate to make for 1,6 bisphosphate or 2. Maybe.
No. Let us talk about glycolysis. In glycolysis, the fructose 6-phosphate, how it will change to 1,6 fructose bisphosphate. How it will change?
By adding kinase.
To phosphoglycerate kinase.
By the phosphofructokinase 1, isn't it?
Yes. Yeah.
Okay, this phosphofructokinase 1 is regulated by a complex process. Can anyone tell me the name of the enzyme that regulated?
Phosphofructokinase 2.
Phosphofructokinase 2. And what is specific about this enzyme?
It is bifunctional.
Bifunctional. Why? Because it has actually two complex. When it's get phosphorylated, it becomes active or inactive. And when it's become non-phosphorylated,
Becomes active.
Okay. But which side become active and which side become non-active? There is two sides in that molecule, or in the phosphofructokinase 2, there are two sides. One side for fructose bisphosphatase, 2,6-fructose bisphosphatase, okay? And this side, when it's phosphorylated, is it active? It's active. And the other side, which is the fructose phosphokinase 2, when it's phosphorylated, it's non-active, okay? We will explain it in this now. So the reversal process for this, we have the fructose 1,6-phosphate in the presence of fructose 1,6 bisphosphatase. So we have bisphosphatase. This enzyme will remove 1-phosphate, okay? And this will give us fructose 6-phosphate. We see that this enzyme, this process is inhibited by the fructose 2,6 bisphosphate and the AMP. However, this enzyme is induced by ATP. So what kind of regulation is this? Allosteric, covalent, or like induction and repression?
Allosteric.
Allosteric, okay. So the third enzyme that's be regulated, which is the glucose 6-phosphatase. This glucose 6-phosphatase will be only present in the liver. And what does it do? It's present in the endoplasmic reticulum of the liver, the smooth endoplasmic reticulum in the liver, where it will take the glucose 6-phosphate and it will remove the phosphate and then will release the glucose again to the cytoplasm, and that make the glucose able to travel from the cell. So this enzyme is present in the liver and the kidney, but not in the muscles. This enzyme of the endoplasmic reticulum membrane catalyze the final step of the gluconeogenesis and in glycogen degradation. Its deficiency results in severe fasting hypoglycemia because free glucose is unable to be produced from either gluconeogenesis or glycogenolysis. So the complete pathway in general: amino acids, lactate, some amino acid will produce pyruvate directly, some amino acid will make it very short and directly produce the oxaloacetate. However, and the oxaloacetate in a reversal step with a detour, that's very important, using the malate, will produce the phosphoenol pyruvate, and then the next irreversible step to produce phosphoglycerate, 3-phosphoglycerate, 1,3-diphosphogylacerate, which will be later producing glyceraldehyde 3-phosphate and dihydroxyacetone phosphate to produce fructose 1,6-bisphosphate, fructose 6-phosphate, glucose 6-phosphate, and then glucose. The glycerol is the source of the dihydroxyacetone phosphate in this process. So this is also another example of the process or the reversal of this process, another cartoon that will make it also easier. We have 2-lactate. This 2-lactate will give us 2-pyruvate. This 2-pyruvate will go to the mitochondria, where they will give us 2-oxaloacetate, and we notice here there is 2 ATP used, isn't it? So the body has consumed 2 ATP, and then here from the oxaloacetate to phosphoenol pyruvate, it also consumed 2 types of energy as a GTP. And then in here, it also consumed 2 types of energy when the ATP changed back to the ADP in order to phosphorylate the 1,6-phosphoglycerate. So from this figure, how many ATP or how many energy phosphate bound has been consumed? 6. Okay, we know that from pyruvate to oxaloacetate there were 2. From oxaloacetate to phosphoenol pyruvate there was 2, and from the phosphoglycerate to 1,3-bisphosphoglycerate there was 2 ATP consumed. Okay. Clear?
Yes.
دكتور، وفي الجلايكوليسيس العكس تماما صح؟
في الجلايكوليسيس العكس تماما. الجلايكوليسيس في البداية بتستهلك 2 ATP، بعدين الناتج بتعطيك 4 ATP وبتعطيك NADH+. ففيه كومبوننت تقدر تعوض. يعني مثلا الجلايكوليسيس بشكل عام تستهلك 2 ATP وتعطي 10 ATP. تمام. بينما في البروسيس اللي احنا نشوفها الحين فيه 6 phosphate or 6 energetic has been consumed. Okay. So the regulation of the gluconeogenesis. This could be a little bit annoying because a lot of enzymes, molecules, and substrate. So the substrate availability, decreasing level of insulin will favor the mobilizing of amino acid from muscles protein and provide carbon skeleton for gluconeogenesis. Glycerol is available due to the accelerated lipolysis in the adipose tissue. The ATP and the NADH coenzymes required for gluconeogenesis are primarily provided by the fatty acid oxidation. So the glucose-alanine cycle, we have mentioned that before. After an overnight fasting, the body as whole is negative. The body will have a lot of negative nitrogen balance. The muscles and splanchnic tissue are the principal site of the protein degradation and release of amino acid into the blood. So the level of amino acid in the blood will be increased. For example, the alanine and the glutamine, which are particularly important, represent almost 50% of the total amino acid released by the muscles, even though these amino acids represent only 10 to 13 of the total amino acid in muscles protein. Okay, but they are the most that's going to be released by the muscles. So simply, during glycolysis, when there is a lot of glutamate, this glutamate will help changing the pyruvate to alanine, and the alanine will travel to the liver, and in the liver, the alanine will go back to pyruvate, and the glutamate will be also produced. The glutamate will go to the urea cycle. Okay, the glutamate will go to the urea cycle, and the pyruvate will be used in gluconeogenesis. So the allosteric regulation of the enzymes of the gluconeogenesis. We know that there are 3 enzymes in the gluconeogenesis. Yeah, can anyone remind me of them? 3 specific enzyme, or 4 specific enzyme for the gluconeogenesis.
الجلوكوكاينيز والفوسفوفركتوكاينيز والبيروفات كاينيز اتوقع.
لا يا شيخ، لا، انا قلت الجلوكونيوجينيسيس. الجلوكونيوجينيسيس there are 4 enzymes that's very important for the reversal to synthesize glucose. انا اتكلم عن العكس، يعني مثلا من بايروفيت الين يطلع لنا الجلوكوز فيه 4 انزيم استخدمت، ممكن احد يذكرني فيهم؟
البيروفات كاربوكسيليز.
البيروفيت كاربوكسيليز، ممتاز. وهذا ايش يسوي؟
اللي هو شو اسمه يعني يحط كربون دايوكسيد باستخدام الاي تي بي.
ممتاز، يعني يعطينا اوكسالو اسيتيت. ممتاز. طيب الانزيم الثاني
الفوسفوينول
بيروفات الفوسفوينول بيروفات كاربوكسيليز.
الفوسفوينول بايروفيت كاربوكسيليز، وهذا ايش بيعطينا؟
كاربوكسيكاينيز ولا
ايش بيعطينا يا شباب؟ من اسمه ايش بيعطينا؟ فوسفوينول بايروفيت. تمام. طيب الثالث، خلاص يا عبد العزيز انت جاوبت لي 2، ما قصرت. ابي اشوف واحد ثاني يجي يعطيني 2 الثانيه. اياد بغازي. اياد نايم. أسامة الحمد.
في حقة الجلوكوز او معليش الفركتوز. نسيت
لا خليني يزيد. انا ابي اسال، ابي اشوف من اللي حاضر معي فعليا ومن اللي فاتح الزوم وقاعد.
اوكي.
أسامة الحمد. أسامة الحمد.
دكتور في فركتوز بسفوسفاتيز او شيء زي كذا.
يا محمد يا أحمري، صح؟ محمد الأحمري. أنا عارف أنك منتبه معي، وعارف أنا أشوف في ناس 3 أو 4 هم اللي يتكلمون. أنا أبغى أشوف، لأن القاعة فيها الآن الروم فيها 97 واحد، أبي أشوف من هو اللي فعليا موجود ومن هو اللي فاتح الزوم بس.
دكتور ناديتني أنت؟
شلون؟
ناديتني أسامة الحماد.
أسامة، كيف حالك يا أسامة؟
الحمد لله.
وش هو التوبيك حقنا اليوم؟
الجلايكونيوجينيسيس.
ممتاز. احنا تكلمنا وقلنا فيه 4 انزيم في الجلوكونيوجينيسيس. شوف أسامة، أنت الحين على عاتقك حماية باقي زملائك، لأنك إذا ما جاوبت بكمل فرع الليستة، فشد الهمة. شكلهم بيغششونك في الواتس الحين.
الله يسمع منك.
فخلك فاتح الجروب حقكم عشان يعطونك الإجابة. احنا ذكرنا قلنا أن الجلوكونيوجينيسيس فيه منها 4 انزيمز. زين، في 2 انزيمز ذكرهم زميلك عبد العزيز، باقي 2 انزيمز ودي اعرفهم يعني.
اللي نستخدمهم يعني
اللي نستخدمهم في الريفرسال ستيبس.
والله يا دكتور
هي واحدة من الثاني، يا ما انت فاهم يا ما انت مركز معي.
لا كنت مركز بالبداية شوي.
طيب وش اللي خلاك ما تركز؟ ما
نفس دخلت بالعميق مرة بعدين شطت شوي.
طيب ركز شوي. اوكي ركز يا أسامة، ركز.
يعطيك العافية.
طيب خلنا نشوف إبراهيم العسيري.
سامي يا دكتور.
ها يا إبراهيم عطنا الإجابة.
الصراحة ماني مركز، أكون معك صريح.
ما أنت مركز ليش يا شباب؟ ما أنتم مركزين. داري، أنا أتوقع أنكم ممكن تسجلون المحاضرة وتقول خلاص خليني الحين أذاكر ولا أسوي شيء ثاني وبعدين أرجع أسمع المحاضرة، بس إنه يعني الأفضل أنك أنت تركز في المحاضرة، تختصر على نفسك وقت أنك تسمعها بعدين. يعني بدال ما تحتاج أنك تسمع المحاضرة بعدين لمدة 2 3 ساعات عشان تفرغها، ممكن ما تحتاج تسمعها إلا ساعة ولا نص ساعة. زين. طيب يا محمد الأحمري، بنرجع لك الحين مرة ثانية، لأن يبدو لي أن في كثير ما هم مركزين. في واحد رافع ايده، خليني أشوف. يزيد الشدي، يلا يزيد.
ما ادري اذا محمد يبي يجاوب عادي، بس جوابي نفس جوابه.
يا محمد ما رد. محمد شكله ها.
تفضل.
اللي هو 1,6 فركتوز بسفوسفاتيز. طيب باقي لنا واحد.
يزيد جاوب ولا أقول؟
لا تفضل.
الجلوكوز، لكن نسيت تكملته.
الجلوكوز 6 فوسفاتيز. فوسفاتيز وليس فوسفات. تمام. طيب أنا والله أفكر نهاية كل محاضرة أعطيكم كويز عشان نشوف من مركز ومن ما مركز. شكلي بصير أعطيكم كويز على البلاك بورد، 5 أسئلة من المحاضرة، بس طبعا مو بالأسئلة اللي بتجيكم في الاختبار، يعني عشان مو بيجي الواحد يقول والله أبى أذاكر الأسئلة هذه وخلاص، لكن أبى أشوف كيف الوقت يساعدني. طيب الأستريك ريجوليشن of the enzyme of gluconeogenesis. عندكم محاضرة بعد هذه؟
خليني أكلم دكتور، ناخذ من 10 دقائق مثلا إذا الوقت.
خلنا نشوف إذا جات الساعة 10 وحنا باقي ما خلصنا نكلم ناخذ منها 5 دقائق. هو عنده 2 ساعة ولا ساعة؟
عنده ساعة.
عنده ساعة. طيب
لكن أعتقد الوقت لأن شرحنا جزء من المحاضرة أمس.
أي ممكن ما يحتاج أكثر. أوكي. Acetyl-CoA activates the pyruvate carboxylase and inhibits the pyruvate dehydrogenase, diverts البايروفيت toward ايش؟ الجلوكونيوجينيسيس. تمام. لما يصير فيه accumulation للبايروفيت، تمام، هذا بيخلي وانهبيشن. لما يصير في عندنا اسيتايل كو اي كثير راح يسوي انهبيشن للبايروفيت كاربوكسيليز، راح يسوي اكتيفيشن للبايروفيت كاربوكسيليز. تمام. ويسوي انهبيشن للبايروفيت ايش؟ ديهيدروجينيز. تمام. وهذا راح يسوي ايش؟ will drives the pyruvate toward the gluconeogenesis. ال AMP will inhibit the fructose 1,6 bisphosphatase while the ATP will activate it. The fructose 1,6 bisphosphatase in the liver and the kidney is allosterically inhibited by fructose 2,6 bisphosphate, which is a level affected by glycogen. تذكرون البروسيس هذه حقت الجلايكوجين؟ اوكي. الالوستريك activation of the hepatic PC by acetyl pyruvate kinase by acetyl-CoA. It's occurred during the fasting as a result of increasing the triacylglycerol hydrolysis in adipose tissue. The liver is flooded with fatty acid. The rate of formation of acetyl-CoA by beta oxidation of this fatty acid exceeds the capacity of liver to oxidize it to CO2 and water, and as a result the acetyl-CoA accumulates and activates pyruvate carboxylase. The single compound can divert pyruvate toward gluconeogenesis and away from the TCA cycle. Covalent modification, this process is regulated by glucagon and epinephrine, which increase the concentration of the cyclin A and B in liver. عارفين وش الcyclin A and B؟
اللي هو الماسنجر.
اوكي هو ماسنجر. لما يرتبط الجلوكاجون بالريسبتور في الليفر راح يسوي ايش؟ اكتيفيشن لايش؟ للسايكلين اي ام بي، والسايكلين اي ام بي بتسوي فوسفوريليشن لبروتين. وشو؟
بروتين كاينيز.
بروتين كاينيز اي، والبروتين كاينيز اي وش راح يسوي؟
فوسفوريليشن
لايش؟
اللي لا اله الا رسول الله، اللي هو البي اي بي 2
الفوسفيت فركتوكاينيز 2. تمام. الفوسفيت فركتوكاينيز 2 لما يصير له فوسفوريليشن ايش معناته؟ بتوقف الجلايكوليسيس ولا تستمر؟
لا
توقف الجلايكوليسيس. تمام. يعني الهدف من هذه البروسيس اللي هو ايش؟ ايقاف الجلايكوليسيس. in turn stimulates the cyclin A and B dependent protein kinase, leading to the phosphorylation and inactivation of phosphofructokinase 2. الglycogen phosphorylates the bifunctional enzyme, lowering the level of fructose 2,6-bisphosphate, which is a potent inhibitor of fructose 1,6-bisphosphatase and gluconeogenesis. تمام. لما يصير عندنا 2,6-bisphosphate هذا راح يسوي inhibition لعملية الجلوكونيوجينيسيس. تمام.
دكتور معناته انه ما فهمت الكوفالنت موديفيكيشن والله.
اوكي الحين هذه الصورة هنا تشرح لك. عندنا الآن جلايكوجين مرتبط، الهاي جلايكوجين مرتبط بالريسبتور. هذا راح يحفز الكاسكيدز اللي هنا او السايكليز اللي هي الادينايليل سايكليز. تمام. وراح يسوي كونسيومينج لاي تي بي عشان يسوي لها اكتيفيشن. راح يتكون عندنا السايكلين اي ام بي. السايكلين اي ام بي راح يسوي اكتيفيشن للبروتين كاينيز. تمام. لما يسوي اكتيفيشن للبروتين كاينيز تمام معناته راح يصير ايش؟ فوسفوريليتد. تمام. لما يصير فوسفوريليتد الفوسفوفركتول كاينيز 2 بيصير ايش؟ inactive. والليفل حق الفركتوز 2,6-بوسفوسفات وش بيصير فيه؟ يقل. تمام. الفوسفوريليشن للفركتوكاينيز 2 domain inactivated and allowing the bisphosphatase 2 domain to be to be active, and this when it's active it will help it will decrease the level of fructose 2,6-bisphosphate and decrease the inhibition of fructose 1,6-bisphosphatase, which lead to increase the rate of the gluconeogenesis. واضح؟ تمام. يعني الفكرة اللي موجودة هنا إن البروتين كاينيز A بيسوي فوسفوريليشن للفركتوز للفوسفوفركتوكاينيز 2. تمام. هذا بيسوي له إيش؟ inactivation. نفس الوقت هذا الفوسفوريليشن of phosphofructokinase 2 will activate it and allow the fructose bisphosphatase 2 إيش يصير له إيش؟ activation. لما يصير له activation راح يساعد في عملية إيش؟ تحول ال 1,6-بسفوسفات إلى فركتوز 6-فوسفات.
معليش دكتور. الهدف من الريجوليشن هذا إن نزيد الجلوكونيوجينيسيس ونقلل الجلايكوليسيس صح؟
نعم.
دكتور يعني البي اف كي 2 لو صار اكتف تصير فيه جلايكوليسيس، اما لو صار الاف بي 2 اكتف يصير جلوكونيوجينيسيس.
إذا صار ال FBB active يصير gluconeogenesis. أما إذا صار inactive يصير إيش؟ يعني شوف هذا الأكتيف فورم حقه. في الأكتيف فورم حقه راح يصير عندنا الجلايكوليسيس. في الانأكتيف فورم حقه راح يصير عندنا إيش؟
gluconeogenesis.
الجلوكونيوجينيسيس. واضحة؟
اي نعم.
طيب احنا قلنا انه البروتين كاينيز بيكون لها تأثير على ايش؟ على البايروفيت كاينيز. ليش؟ لانها بتسوي له فوسفوريليشن، وفي حال سوت له فوسفوريليشن راح يصير ايش؟ inactive. فالفوسفوإينول بايروفيت ما راح يقدر يتحول لايش؟ لبايروفيت. اوكي؟ So الجلوكاجون phosphorylate البروتين kinase which causing it to be inactive. So diverting the phosphoenol pyruvate to the synthesis of the glucose. يعني من ضمن البروسيس اللي يسويها الجلوكاجون إنه راح يسوي عن طريق الactive protein kinase A, it will phosphorylate the pyruvate kinase, and this phosphorylation will lead to inactivating it and allow the pyruvate to go back to oxaloacetate and then pyruvate enol and then phosphoenol pyruvate. Okay, induction of enzyme synthesis. الجلوكاجون increase the synthesis of phosphoenol pyruvate carboxykinase while insulin decreasing it. Gluconeogenesis and glycolysis are reciprocally regulated. That's one pathway is relatively inactive while the other is highly active. يعني ايش reciprocally؟ يعني reciprocal regulation، يعني إذا واحدة active الثانية تكون inactive، يعني يتبادلون ايش؟ أدوار. الفركتوز 2,6 bisphosphate plays an important role in determining whether glucose is to be degraded or synthesized. Okay? عارفين الفركتوز 2,6 bisphosphate؟
يعني كميته هي اللي تحدد
كميته. لإيش؟ لأنها allosteric regulator.
هو اللي يتحكم بالبي اف كي 1 صح؟
ممتاز، هو اللي يتحكم بالبي اف كي 1. نعم. الريسيبروكال كنترول between gluconeogenesis and glycolysis، الالستريك ريجوليشن، الاسيتايل كو اي والانديوس the pyruvate carboxylase. الاي ام بي will inhibit the 1,6 bisphosphatase, fructose bisphosphatase. However, the ATP will induce the 1,6 bisphosphate. Fructose 2,6 bisphosphate will inhibit the fructose 1,6 bisphosphate. Okay, increasing the glucagon will cause the reduction in the insulin glucagon ratio, and it will decrease the fructose 2,6 bisphosphate. Also, it will induce the phosphoenol pyruvate carboxykinase, and then it will activate the gluconeogenesis. So in summary, gluconeogenesis is the synthesis of glucose from non-carbohydrate anabolic, energy consuming. Four unique enzymes are required for reversal of the three irreversible reactions of glycolysis. Both gluconeogenesis and glycolysis are reciprocally regulated. تمام؟ بكذا تكون انتهت محاضرتنا اليوم. عارف في جزء في المحاضرة عديناه بسرعة. بعد ما تسمعون المحاضرة شوفوا شي أسئلتكم وارجعوا لي. ممتاز.
اوكي بإذن الله.